something that I wanted to know about, so selfishly I'm excited for this presentation because I've seen it in Humboldt now, and I've seen some of his adventures on Facebook, and now I get to hear the whole story. So basically, Terry went to grad school in Wyoming, and during that time, or just before that time, he was on a Smithsonian flora project in Guyana, and that brought him to the tropics for the first time. And he followed that up with a PhD at Duke, and now he has been at HSU for how long? I should ask you this.
Since 2002.
Since 2002, he teaches forest pathology, which is where I met him as a student about 6 years ago. Really opened my eyes to the forest in whole new ways. It's amazing relationships he was able to share with the class. But he also teaches general mycology, fleshy fungi, and plants and civilization. So with that, I'll introduce Terry Henkel and look forward to the talk tonight. Thanks, Terry.
Thanks to Michael, too, for recruiting me to give a talk. I gave one CNPS talk a while back, but it might have been the better part of 10 years ago. One of my memories, besides the cool interactions I had with the Plant Society members, was all of our computer system and everything broke down that night. And at one point I had to do some damage control and run up to HSU and grab a projector and a computer, but we finally got it going. But I think we got out of here about midnight. But we might not get out till midnight tonight. No, I'm kidding. I'm going to start my talk tonight by actually showing a video, 4-minute clip that my former grad student Jessie Euling made, who's now doing her PhD at Duke, who did a lot of field work with me in Guyana. I'm not going to say anything about it, just watch it for 4 minutes. Give you some idea what our field work trips are like in the most remote rainforests of tropical rainforests of the world. if you want to know anything about the details of what you just saw in the video, including the botanical identity of that black gunk that I was sticking in my mouth, you can talk to me after the talk. Fair enough? Or where we were and stuff on that expedition. So, my talk tonight is going to be, by nature, fairly mycocentric, mostly on fungi. But there'll be some botanical elements in there. And in the end, especially the group of fungi that we work on who are mutualistic symbionts with higher plants as mycorrhizal fungi, there's always the element of fungal and plant interaction. They're not happening in a vacuum in the environments that they live in, etc. So I'm going to review some of the research results along the lines of fungal forest habitats in Guyana, the Guyana Shield region of South America, and how particular host trees, mycorrhizal host trees that are unique to the region, support a really vast community of symbiotic fungi that wouldn't be in these ecosystems unless these fairly rare lineages of woody plants were there. And run through a bunch of stuff related to that, and then toward the end, talk about what I was able to find on my first excursion into tropical Africa, which I did this past year on my sabbatical year, in the country of Cameroon, in the Congo Basin in Central Africa, and why we went over there, and what we're trying to find out over there after working for so many years in the New World tropics, how that all fits in. I know at the end of the talk I'm not going to have time for acknowledgments, so I'm going to put up a big acknowledgement sign at the beginning, but not say too much about any particular individuals in here. We've been working, especially in Guyana, and myself and colleagues and graduate students of mine, et cetera, and others now for the better part of 20 years. And there's been a lot of contributing individuals from a research standpoint, funding agencies, field assistants, et cetera, that are too innumerable really to mention by individual names. But I put some of the key players up here. Now, where's my laser pointer? Oh yeah. Cool. We've had funding from several major institutions, National Geographic Society, National Science Foundation, and others. The individuals that I have in blue font have been especially strong contributors in various ways to the work over the years. All of those listed under the different categories have done something substantial. But particularly Kathy Aim at Purdue, my close colleague, Dave Largent, who's still here in Humboldt County, and has worked up a whole lot of our fungi in his taxonomic group in Guyana. We've published 6 or 7 papers now as far as collaborating taxonomists. Field logistics, Mimi Chen, she's here today. Certain grad students, of which all of them have done projects of some kind in the Guyana system, but in particular whose work will be featured in a couple of places in this talk. Jesse Ewing, Marcia Parkey Group, who just finished, and Rachel Koch, who is a grad student at Kathy's at Purdue. And Matt Smith and Rita Spilbelies as colleagues in the molecular ecology components of the work. Well, ectomycorrhizae, just some general topic outline. Why are they important? What are they in the first place, and why are they important? We're going to talk about tropical tree diversity, monodominant forests, and ectomycorrhizae, and what's the big deal about all these things. The unique geographic and phytogeographical position of the Guyana Shield region of South America. And what do we know about ectomycorrhizal fungi in the tropics? Is their conceptual disjunct. We'll see what I mean by that as we go along. What can we learn from a site that we've studied to death? We've been working now in the same field system, as remote as it is in Guyana, and monitoring and working in the same plot array for the better part of 15 years now. What can you learn about studying a system enough to really get some of the details on biodiversity presence, fungi in this case, that you could never get from doing like a one-off field expedition or a one-off visit to it to collect some kind of data or specimens. Fungal, ECM fungal hyperdiversity in Guyana is assigned before us, how does tropical Africa fit in? Now, ectomycorrhizae, there's an example of an ectomycorrhizae. It is a type of mycorrhizae. Mycorrhiza are the default position for plant roots in nature. Very few plants in any natural environment on the planet Earth exist in a non-mycorrhizal state. They always have, with a few exceptions in certain families, like Brassicaceae, for instance. They form some kind of symbiotic relationship with some kind of fungus in the soil where they trade carbohydrates to the fungus for the fungus's carbohydrate nutrition in return for a greater explorational range in the soil for mineral nutrients and water. Classic mutualistic symbiosis where each intimate partner in the relationship is gaining something from being in the relationship. Ectomycorrhizae are a type of mycorrhizae that are formed from the fungus standpoint usually by higher basidiomycete fungi, mushroom-forming fungi, and some astromycota. They are not the most prevalent mycorrhizal type in the world. The arbuscular mycorrhizae are a completely different fungal group. And the number of plants overall, terrestrial vascular plants that engage in the arbuscular mycorrhizae relationship is much higher if you look globally, the number of families, genera, and species. But the actomycorrhizal symbiosis occurs in selected lineages of woody plants, both gymnosperms and angiosperms. And although the total diversity of the actomycorrhizal plant groups viewed globally is not nearly as high as the amount of plant groups that do arbuscular mycorrhizae, in many ecosystems, like ours, right around here, most of our forest types are dominated by ectomycorrhizal trees. Members of the Pinaceae, members of the Fagaceae. That situation changes when you get into the tropical rainforest, and we'll get into that in a minute. But this type of mycorrhiza has evolved independently multiple times in the fungi across different lineages of the basidiomycota, and evolved independently in the plant groups who are often phylogenetically disjunct from each other, the Fagaceae, the Pinaceae, the dipterocarpaceae in Southeast Asia, independently settled on this type of mycorrhiza as their root symbiosis system. And because of that, the fungi that are in those groups have evolutionarily radiated into a lot of diversity. And to some extent, radiations have occurred in those particular plant groups. Think about the oaks, for example, which are quite diverse, Quercus and allied genera of the Fagaceae. In many, including tropical, if you look at Southeast Asia, forested regions of the world, the Fagaceae are quite diverse and prominent players. It's an actomycorrhizal plant group that's radiating. Approximately 7,000 actomycorrhizal fungal species that are currently described in about 250 different genera. But all the various sorts of extrapolations about how much do we know about fungal diversity, how much is undiscovered versus already known and described in the actomycorrhizal groups. The estimates are that their diversity of ECM fungi worldwide may exceed 20,000 species out of 50,000 macrofungal or mushroom-type fungal species in the world as estimated. So that means the majority of them, if those numbers are anywhere close to correct, are undiscovered still. And they're vastly important ecologically, actomycorrhizae, for reasons we'll get into as we move along a little bit. So geography here. Northern South America, our sister continent to the south of us in the Western Hemisphere, has the largest contiguous expanse of lowland tropical rainforest. It did originally and still does today, even with human-driven deforestation. So in this great lobe of northern South America, there's a huge watershed, the Amazon River watershed, that geographically encompasses pretty much all of this territory. If you took the lower 48 of the United States and plastered it down on here, it would fit with room to spare inside just the Amazon Basin watershed. That's how big it is. It's the largest river in the world. It's the longest river in the world, etc. And it is the tree diversity motherlode of planet Earth, the Amazon Basin in general. All tropical rainforests are diverse for plants, and especially diverse for woody plants, but you get the most diverse tropical forest in the greater Amazonia, particularly in the western Amazon, where you can have single 1 hectare, that's 2.2 acres, plots with over 200, and some plots have been measured with 300 or more different tree species in 1 hectare above 10 centimeters in diameter, which is a DDH about like that, in 1 hectare. That's over in here, and mostly in the central Amazon basin. But in the greater South American tropics, there are regional variations in the plant community composition and some aspects of diversity of trees in the forest, including areas where you have dominance of individual sites by single or small groups of tree species, which otherwise is pretty much very rare in tropical rainforests. Our plant communities around here, as far as trees go, you can walk into a 4,000-foot elevation mixed Doug fir, white fir, and if you know your woody plants at all, you will have identified all the woody plants in your immediate vicinity in about 5 seconds. Maybe 5 different species, a couple of hardwoods, a couple of dominant conifers, done. Those kinds of situations can occur in the tropics, and we're going to get into that because the system we've been studying in the Guiana Shield region has examples of tree dominance in the forest, and it ties directly into the fungi in a way I'll make clear. But this is the Guiana Shield. The countries Guyana, Suriname, French Guiana, Eastern Venezuela, and this northern territory of north-central Brazil. It's geologically unique. It's a shield region, Precambrian basement igneous rock. It's the only shield in South America. And it has scattered pockets of sandstone overlay here where you see mountains in central and eastern Venezuela coming into Guyana. So the Guiana Shield, Guyana Highlands region is unique relative to overall Amazonia in terms of the geology, the phytogeography, and the phytosociology, how the tree communities compose themselves or assemble. Now, I've been working in a big collaborative project with Hans Verstegen and 125 other collaborators, all of which get on the paper as co-authors. It's the most ridiculous example of too many authors on a single paper. Our most recent that's published was this hyperdominance in the Amazonian tree flora in Science a couple of years ago. We have a plot network now that actually is bigger than this. We're up to like 1,400 plus 1 hectare fully mensurated tree plots where everything's measured and identified, scattered pretty much basin-wide in the Guiana Shield across the South American tropics. And what Hans and the main collaborators have been doing is crunching these data in all kinds of interesting and creative ways. So this paper was called Hyperdominance in the Amazonian Tree Flora. We just learned that most tropical rainforests don't have dominant tree species that you can recognize because they're hyperdiverse, a lot of species packed. This was a crunching down and metadata analysis of all these plots in Brazil, Peru, Guyana, Venezuela, Colombia, Bolivia, etc. And by looking basin-wide, some things emerge from this, and this is a complicated diagram, but the things that I want to highlight, I'll say briefly. It's estimated that there are about somewhere on the order of 15,000 plus different tree species in the greater Amazonian region. That's a pretty high number. But looking at a meta-analysis of all these plots combined across the basin, out of those 15,000 plus species, most of them that would be considered extremely rare in any kind of sampling event. 227 species estimated to make up over 50% of the individual trees across the Amazon basin. What this general result means is that pretty much if you go in and lay down a plot in the lowland South American tropics anywhere, measure off all the trees and identify them, it's quite likely that you're going to come up regularly in repeated censuses of the trees with some of these 227 species that figure prominently in the basin-wide data set. And then there's a gazillion other trees that would be considered extremely rare and unlikely to be encountered but have occurred in a census here or a census there in the data set. So I'm trying to bring this down. What Hans was trying to get out with the hyperdominance is that when you look on a broad scale, there's a small assemblage of tree species that are widely distributed and likely to occur in a sample that you lay down anywhere in the Amazon basin. It doesn't mean in that sample they will be dominant at the site, like every other tree is a single one of these 227 species, not that. Some of the conclusions that from crunching these data from the Amazon plot network, though, that show how the Guiana Shield region, this northern part of South America, is unique relative to the rest of the Amazon basin, are some things like within the woody plant community, there's a very much higher proportion of fabaceous woody plants when you get into Guiana Shield compared to the central or southern Amazon. Now, we have a weird, skewed concept of legumes as temperate zone people. Most of the legumes that we're familiar with are herbaceous. There's some woody ones, yeah. There's Rhodania, pseudoacacia, a few oddball tree legumes. You get into the tropics, especially afrotropics and the neotropics. Almost every other tree you see in the forest is a giant canopy tree that's a fabaceous tree. Fabaceae rule the neotropics and the afrotropics as a family. And we're not used to seeing giant canopy trees in the Fabaceae, but they exist and they're very numerous across many genera and taxa in the three subfamilies in the tropics. They're even more numerous in the Guiana Shield. Diversity on a plot or stand level of woody plants is way lower in the Guiana Shield in general, like 30 to 80 species per hectare, these smaller black dots versus these gigantic dots, the biggest ones being 270 to 300 species per hectare over in western Amazon. So lower diversity, a lot more Fabaceae, especially Cs alpinioid subfamily. Low disturbance regime. The trees have bigger seeds that often have gravitropic seed dispersal mechanisms. The legume pods just open up, the seeds go boom, right down on the forest floor without any specialized dispersal mechanisms. And they actually often establish seedlings in close proximity to the parents, which is unusual in tropical rainforests because of host-specific seed predators and pathogens. So that part's kind of weird. Wood density tends to run higher in trees in the Guiana Shield. So some of the most dense woods in the world are from Guyana. They're like pieces of iron when they're dried out and milled into lumber. All this points to, as a regional characteristic, we must have had rainforests, tropical forests in the Guiana Shield for a very long time without any major shifts due to climatic change and no regular large-scale disturbance factors causing constant disturbance and reassociation. So stable equilibrium forests that have shade-tolerant seedlings of trees that are able to recruit close to their parents. That's weird for tropical rainforests, trust me. So we get what we have found and was noted earlier that when you get into the central part of the Guiana Shield, like the country of Guyana, you can find distinctive looking tree communities where you can walk in them repeatedly. Mid-slope and hill country, you're always going to find 2 or 3 canopy trees that are abundant in that stand, and you recognize that, yeah, this is that forest type. There's a Morrigan grippii and Eperua. We always see them in association, just like we always think about here. On south-facing aspect, it's going to have incense cedar, Jeffrey pine to go over the side. On the east, it's got a more amazing forest, different tree composition. This you don't see in typical lowland tropical rainforests. It's just a constant mishmash of gobbledygook of tree diversity, and you can't see any pattern in the landscape often. Not true in Guyana. So that was noted in the lowlands. And what we started observing during my Smithsonian days in the mountains of western Guyana, in that sandstone country, were pretty extensive stands of forest that were completely dominated by a single leguminous tree species. Over fairly substantial acres, patches of these old growth stands, all the forest in the area is old growth, that are 4 or 5 or 10 hectares in size, where it's very, very continuous. One dominant canopy tree species, and the seedling and sapling recruitment pool is predominantly the same species. So monodominance is what I'm getting at. These are in leguminous plants in the genus Decime, which is
endemic genus to the Guiana shield in the Caesalpinioideae, one of the 3 subfamilies of the basin. So here's an aerial shot from a small airplane flying into our field site in the Upper Pataro River Basin in that mountainous region of western Guyana. There's the river, there's a nice contiguous forest canopy, there's some low mountains in the background. And by looking at that, yeah, it's big, substantial forest. It could very well be very diverse. But this same area, this is a figure from one of our remote sensing assessment papers of this area of forest in western Guyana. Rebecca DeGagny was a grad student here doing GIS work that worked with me on this a few years back. This is about a 20-kilometer on the side piece of this landscape. And based on ground truthing, ground-based measurements in selected sites and analysis of Landsat imagery from that you can download of this region from Landsat and analyze the spectral signatures coming off the canopy of the trees, we were able to figure out what a Desimbe monodominant canopy looked like based on its spectral signature in the Landsat photographs by combining known sites where we had ground data and doing a broader analysis across the Landsat image. So everything that's in green there is estimated with about an 80+% probability of being true is Desimbe monodominant forest. That's 20 kilometers on a side. And the yellow is basically some type of mixed forest, and the dark green a type of swamp forest that's dominated by a euphorb canopy tree. So we were able to actually pick out these things from satellite imagery and get better estimates of how extensive these monodominant forests were in this area. It's something that's very hard to do in tropical forests in general because of the usual extreme heterogeneity of the forest canopy, not allowing you to pick up pattern in plant community composition. So this tree, the Desimbe corymbosa, it's in the Caesalpinioid subfamily. It's close, it has a multiple stem regeneration, root adventitious regeneration that leads to large multi-stem, highly complex reiterated trees that don't look like a normal tree with a single trunk and one crown at the top. This is an extreme example of a very, very old Desimbe that has reiterated on top of itself for many centuries. Its closest relatives, I told you that the Fabaceae was the dominant tree family in the New World tropics. So this Desimbe is in the New World tropics. It's in the Caesalpinioideae, but its closest relatives phylogenetically are a group of Caesalpinioids that are otherwise restricted to tropical Africa. We know this from phylogenetic studies in the Fabaceae, and all of those that are in tropical Africa that have been studied do ectomycorrhizal symbiosis. The Desimbe, the only known New World member of this particular lineage of Caesalpinioids, the tribe Amperstidae, is neotropical and does ectomycorrhizing. Many of the African ones form monodominant forests, and this one in the New World forms monodominant forests. This is, you can keep that in mind when I get around to the justification for finally going to Africa last year.
So is that,
yeah,
there's a lot of damage and these leaders are always dying?
Well, that's a very complex question. There's turnover in the leaders, and already in place, reiterated shoots that go up into the canopy gaps and add over time to the physiognomic complexity of the individual tree. And there's adventitious root currents coming down to actually join up with the soil and become woody and form what we call a pseudo-trunk when they're really old and highly reiterated, where the support system of the tree is pretty much a massive fusion of woody roots that started out as adventitious roots growing geotropically down that emerged out of the side of the tree at an earlier stage. They're ectomycorrhizal. So we spent a lot of time, and we're still studying the life history traits that we can suss out of Desimbe corymbosa that might contribute to its ability to achieve like 80 to 90% basal area in significant stands of forest and complete dominance of the seedling and sapling pool. How do you create a natural monoculture of a single tree species in the tropical rainforest, where otherwise you'd have hyperdiversity of trees packed into the same area. They've got to get around the mechanisms that levitate against trees recruiting juveniles of their own kind right in close proximity to the parent trees. That's rule number one. Because in all forests, but especially in the tropics, seedlings usually don't get established in a close physical proximity to the parent tree, because there's a whole crop of host-specific seedling pathogens that are maintained at some level on the parent tree without causing mortality, that keep the inoculum potential high, so the seedlings that don't disperse very far away from their parent tree die in the seedling stage from host-specific pathogens. You got to get around those mechanisms to become a monodominant tree in the tropics. So escape from what are called the Jansen-Connell mechanisms. And as the system develops and becomes increasingly packed with individuals of the same tree species, other tree species are competitively excluded from the system if they have a different mycorrhizal type, is what we're beginning to conclude. So with the corymbosa, it does all kinds of stuff that help it perpetuate itself on an individual level and with sexual recruitment of seedlings in the same stand over and over and over to keep it packed with itself. The ectomycorrhizae short cycle the mineral nutrients. The litter falls down, leaves and stuff from the Simbi trees. They're quickly colonized by the hyphae coming off the mycorrhizal roots of the same tree. And they, in a nutshell, they suck the important mineral nutrients right back out of the leaves and pump them back into the plant that just shed those mineral nutrients with the litter fall. So that can keep the mineral nutrient cycling really tight within the domain of this single species on one side. Common mycorrhizal networks might come into play. There's a mycorrhizal community of hyphae in the soil that may immediately join up with new seedlings and allow those seedlings to share nutrients or obtain nutrients in the early growth stages from the parent adult trees that are right around. The complex physiognomy, this epicormic reiteration of shoots and roots, leads to a lot of places where litter gets trapped on the trees. Leaf piles build up, organic soils develop, adventitious roots go in there and mine out the leaf litter above ground in these aerial litter piles. And they also, to top it off, which is why this tree is one of the most super tree examples of any tree in the world, even more than redwood, is because not only does it have this extreme clonality of individuals having no determinate lifespan, because one stem dies, they reiterate new ones, more adventitious roots coming out, they get more complex, more litter trapped. We have no idea how long these things can live, but it seems to be indeterminately. Redwood does that too. Vegetatively, it sprouts up, and the main log gets cut, fire damages it, it resprouts, it has ample reiteration, but redwood does not reproduce by seed very prolifically. You gotta have just the right microenvironment, post-fire alluvial soil deposition, seed fall to get new seedlings. Forget it. With the Simbi, every 4 or 5 or 6 years or more, it goes through an explosive production of fruits. Massive numbers of fruits and seed production within stand, and it drops thousands of seeds, the great majority of which successfully germinate and establish in the stand, so it keeps the recruitment pool stocked for a very long time in the future, when one of those seedlings might actually make it up into the canopy and become one of these large reiterated trees. Almost certainly a process that takes many centuries. So we did, this is kind of a summary of our general working hypothesis of how these trees become dominant in a site and maintain that dominance. Several citation papers we published. So if the ECM trees dominate, well, they're ectomycorrhizal trees. There must be ECM fungi there, right? Yeah, of course there are. You've got to have the mycobiont if you've got the phytobiont in a particular type of mycorrhizal system. But this particular group of fungi was thought for a lot of years to be very poorly represented in the tropics, if not completely absent. So, ectotrophic forest. We're surrounded by them all over the place. in the temperate and boreal zone because most forests have a fairly strong pinaceae component or fagaceae, sometimes both. For instance, two major temperate tree groups that do actomycorrhizae. So an ectotrophic forest, closed canopy tall forests that are dominated by ECM trees. All over most of the northern hemisphere, temperate boreal zones, parts of the southern hemisphere, the nothofagus zones of southern South America, the eucalypts in Australia are ectomycorrhizal, and related genera in the Myrtaceae, as well as eucalyptus. In the tropics, there's some known and some not known. In some places, Southeast Asian tropics, there's a big woody plant family called the dipterocarp, 500 species roughly, that actually as a group often dominate areas of lowland rainforest in the Southeast Asian tropics. Get over into Africa, mostly high diverse mixed forests, but certain areas where these caesalpinioid legumes dominate forest and they're ectomycorrhizal. And then the emerging sites, these are oak zones here in Central America and montane, so they don't really count as being lowland tropical. In the Guyana, greater Guyana Shield region are the only known examples in the lowland neotropics where you get ectotrophic forests. They're dominated by ECM trees. Big deal. But from a mycology standpoint, and I was taught even when I learned mycology early on, that yeah, we got a lot of ectomycorrhizal plants in the temperate zone, but you just don't see them when you go to the tropics. And that's true. You don't see the fungi that do ectomycorrhizae in a lot of tropics because the trees aren't there in many of the specific locales to host them, because the lineage of trees that do it in the tropic are very few. But in areas where they occur, they often dominate the forest. And early mycorrhizal surveys in the lowland tropics, all the trees came up in a particular stand as being arbuscular mycorrhizal. That's part of what created this dog. But if you look at the taxonomy literature, fungal species described from like tropical Africa, some parts of the neotropics, there's a lot of records for things like Amanita, various boletes, russula, which are in fungal groups, genera, and families that we know already or always have to be an ectomycorrhizal situation to occur. So there must have been ECM plants there if somebody collected these Amanitas in the Congo back in the 30s. There was a lot of that literature out there, but no ecologists never paid attention to that. So, and the examples where the forests are dominated by these ECM trees have been largely overlooked where they were known to occur or not discovered until recently. And many regions still remain unexplored. So how we began to rectify the situation in Guyana, which is a country that's situated right in the middle of the Guyana Shield region, we set up a long-term plot study to look at the mushrooms in Decyme-dominated forest and adjacent areas of mixed forest that lacked ectomycorrhizal trees. We monitored those plots with regular visits over a multi-year period in the 2000s. Same time of year, rainy season, a systematic way to sample sporocarps and record morphospecies present and provide a lot of collections to do subsequent taxonomic work. Because as you'll see, most of the things that we find down there are species that are new to science. We have to also describe them formally to get names on them. So in 6 1-hectare plots, 3 in the Decyme forest in this region of the upper Congo Basin, this is that GIS or that Landsat analyzed image. You know, the green is Decyme, are 3 plots, 1-hectare plots gridded off into smaller subquadrats, and 3 in the mixed forest, essentially lacking in Decyme, being close by, within a kilometer or 2 of the other Decyme plots. And went back and sampled those puppies from like the beginning of June till the middle or end of July, intensively for basically 8 years in a row. We've still gone down there and sampled, but not the systematic, repeated visit stuff. We had 8 years of data as of 2007 and worked it up. And I'm not going to present all those data because we're looking at saprotrophic decomposer fungi too. We have like well over 1,500 different species of macrofungi in those 1,600 hectares, most of which will take several lifetimes to do all the taxonomic work on to get them described. But in the ECM fungi, they were all restricted to the Decyme stanza, even on a local basis. It makes sense because it's the only known host tree at that site. We had, in the plots themselves, with 8 years of sampling, 27 distinct species within the plots. scattered across mostly Agaricomycetes, the Basidios, and a few Ascomycota. At the family level, you see a lot of names that if you know a little bit about mushrooms, you're going to recognize. Cordyariaceae, Russulaceae, Ammonitaceae, etc. And most of the genera that we find down there are also genera that you could find if you went collecting in our area up here. Is at the species level where things are fundamentally different. And by sampling the mushrooms repeatedly in the same spot during the rainy season year after year, you realize that there are some species that fruit more regularly, so that they occur more in your data set that from a fruiting body standpoint dominate the system. These are some are boletes, some are ammonitis, some are coral fungi, some are in the cantharelloid group, etc. But we're also collecting at this particular site, general collecting, foraging all the time outside of the plots. And this area, which is really about a 10-kilometer radius area of remote forest in these mountains, we've been collecting intensively, and either in the plots or outside. If you count up all the stuff that has occurred in ectomycorrhizal groups, so we've got outside the plots that never occurred in a plot sampling, you got to add at least 47 more species. And this is where David Largent comes in some, because this genus, Entoloma sensu-stricto, and its allied genera that are in the Entolomataceae are really diverse down there at the species level. And everything that we've collected so far, every single one of them, and we're up to about 40 species of Entolomataceae that we've worked up and described. All of them were new to science, the species. Some other genera that I had no idea you could even expect to ever find in the tropics, like Phaeocolybia. We found a couple of different species of Phaeocolybia, etc. So if you add the off-plot taxa in this local area, about a 10-kilometer radius, we're up to 174 species. This is not an undiverse area. It's in the tropics for ectomycorrhizal fungi. This compares favorably with other plot studies that have been done in the tropics, which basically are just two, and many, though, that have been done in temperate boreal systems. If you set up these hectare plots out here, like in the Doug fir-dominated stand, and went back every year for 7 weeks and counted up and identified all the sporocarps that fruit for like 8 years, you'd probably come up with around 200 species of ectomycorrhizal fungi in the same unit planted. So it's as diverse as a temperate site, which kind of makes sense because it's dominated by ECM trees, a type of system that's very poorly known for the tropics. So if you count up all of the ECM ectomycorrhizal fungi that are already known and described in the literature from the New World tropics somewhere, you come up adding all these disparate publications, you come up to about 150 to 200 species. Our work in this one particular forest in Guyana come up with 174 species. Most of those are species that are new to science, at least 60 to 70% of them. And we've described somewhere about 100 of them, maybe up to 120, I've lost count formally, that we've described in publications, put new names on, and etc. Let's look at some of these mushrooms. So most of them in the different generic level groups, like Russula, like Ammonita, like the different bolete genera, are species new to science. Here's some examples. Boletellus fiacaeti. It's a bolete genus, Boletellus. This was a new species in that group. There's an Inospe, Lilacinus formosa, new to science. There's an Entoloma that we described with Dave. There is Lactarius and Russulaceae or Lactifluus is a new genus now. New species. How do you get names on these things? You don't just cook them up and say, I'm going to throw a name on it. It's a formal process. It takes a lot of work when you get the specimens back from the lab. Detailed micromorphological analysis, DNA sequencing, etc. And then a perusal of all of the world's known literature in the group to make sure that this species has not been described from somewhere else and already has a name. Same thing with plants, but doing basic alpha taxonomic work and bringing it to fruition in terms of publication is a lot of work, and it doesn't get a lot of credit by theoretical or question-driven biologists. But you know, most biologists dream of maybe describing one new species their entire career, if they ever describe anything. We're swimming in it. Mycobarium up at Humboldt has several lifetimes of new species. Somebody can keep cranking this alpha taxonomy out. We're trying, though. several, aside from Dave Largent, several other key taxonomic collaborators that are experts in certain groups, like Brandon Matheny at Tennessee. Ionosity is his genus. Some groups are rarely collected in the tropics. If you know mushrooms around here, Ionospes are super common. You get sick of seeing them during the wet season. They're diverse, sometimes hard to identify, etc. They're not common in tropical rainforest where active mycorrhizal trees are around, but we're up to 11 species down in Guyana, and 9 out of those 11 were new to science. Weird morphologies are rampant. We're talking mushroom and fungal fruiting bodies here. This is a lactarius. These are the milk mushrooms. Around here, every lactarius you ever find has got a big, nice central stipe and a nice umbrella cap and gills. We get members of that family, the Russulaceae, that form these tiny little fingernail-sized fruiting bodies that form in clusters on the sides of trees. And you can tell this is a lactarius immediately in the field if you break the tissues, because it exudes the milky latex like a typical lactarius. But it has this pluritory fruiting body. And this one was described by Singer from specimens in Trinidad. It was the only one known in that family with that morphology until we found several other species that were different in lactarius, and actually one Rushla, that have this pluritory growth form, highly reduced diminutive fruiting bodies that were new to science that we described. What they do, though, is really weird ecologically, these pluritoid lactarius species. All this fuzzy-looking stuff, there's the base of a Decimbe tree and a bunch of Decimbe seedlings and a bunch of leaf litter. All of this fuzzy stuff is what we call a subiculum of one genetic individual of one of those lactarius, the brown Lactarius brunelis. So you find these communities above ground of the mycelium, the fungus. It's also below ground, connected to innumerable mycorrhizal root tips, feeding off that and growing up the trees in these communities of dense hyphae to make a fruiting platform for those little tiny mushrooms to periodically come out and flush out. So it's like an aerial development of mycelium above ground to provide a favorable fruiting position for efficient spore dispersal from those little tiny mushrooms. Very weird. Same genetic individual. This is an Elaphomyces. It's a common false truffle genus in our region. Below ground, animal dispersed, macrofungus, etc. We found this thing a while back and described it initially in 2000, and then did some further work on ecology and stuff we can see in a minute. This is Pseudotelostem. All Elaphomyces known in the world are completely enclosed all the way to maturity. They're designed to attract small mammals, to dig them out of the ground, carry them away, eat these perennial tissues, and scatter the spores as a dispersal mechanism. We found this thing, which took a lot of molecular-based sleuthing to figure out in the end that this was the weirdest Elaphomyces essentially in the world. We put it in its own genus because the morphology is so different. It's an Elaphomyces all the way up to this point where at maturity it pushes the dry spore mass up on a stalk and it breaks through the peridium, leaving a vulva-like structure. And then the spore mass just sits there and waits to get battered by raindrops in the rainy season. It just knocks off clouds of spores until they're dissipated. We now know, now that's a phylogenetic tree, you don't need to pay too much attention to detail, but I'll tell you that I hypothesized early on, but we didn't have the depth of molecular sequence data to figure this out, that in this case, I hypothesized that Pseudotelostoma, with this pushing out of the spore mass, was a derived condition from Elaphomyces truffle-like ancestry. We now have sufficient molecular data. This is a huge phylogenetic tree of Elaphomyces species that clearly shows Pseudotelostoma is nested within Elaphomyces. We have a manuscript we're getting ready to submit describing new Elaphomyces species from Cameroon, but in that paper, we're also going to finally, and much to my dismay, but I agree that we need to do it, eliminate the genus Pseudotelostoma and put it just in Elaphomyces because phylogenetically it's nested right inside the genus, even though the morphology is so different. These are some of the impacts that molecular systematics data can have on your concepts. That's my name I gave it that genus name, and pretty soon when published the paper, that name's not going to exist except it has an early synonym. This truffle macromorphology. They're truffle-like fungi in most ecosystems. We have plenty of them around here, and most of the time, when you figure them out phylogenetically, they're derived from like mushroom ancestry. They've evolved along that trajectory to depend on animals for dispersal instead of shooting their spores off in the gills of the mushroom. But this group, the Physolacreaceae, which is a gill mushroom family that has the genus Armillaria in it, which is a major tree pathogen in our area, all over the world. They're root parasites, and they make the honey mushroom fruiting bodies when they fruit above ground. We have some armillarias in our system in Guyana. They cause root pathologies. Trees tip up early. The armillaria fruiting bodies can come out of the decaying wood after they've killed the tree. Well, we've started finding this black truffle for a long time in Guyana, and morphologically, we had no idea what it was related to. We had to get several loci DNA sequence data, and the pictures started emerging that its closest relative was the genus Armillaria. This Guyana mycorrhizal. We erected a new genus, because at the time we described the taxon, it was not definitively clear that it was nested in Armillaria. It looked like it was sister to it, and with this radical different morphology, it's got to be a new genus, and I made it a new genus. Well, it's also weird from a biology standpoint, dispersal biology, we're trying to figure it out. It looks like instead of attracting vertebrates, like rodents, to drag the truffles around, consume them, poop out the spores, or carry them to a distant site, that it is probably designed for dispersal by invertebrates, particularly termites. Because as those very dense truffle-like fruiting bodies mature over several weeks, they invariably get excavated by termites and a few other invertebrates, but usually lots of termites, just gobbling up that red spore mass in the tissue in the middle. And they do gobble it up. There's one under the second scope, a fairly clear body, and its entire alimentary canal is packed full of this red leaf material, which has a lot of spores in it too. There's a spore that was squeezed out of the gut contents of one of these termites, intact. So we think that this thing is not only weird, it's the first known truffle in the Armillaria group to ever be discovered, but it also looks like a truffle-like enclosed fungus that's designed for invertebrate excavation and spore dispersal instead of attracting vertebrates or mammals to dig them up and carry them around to disperse. We're still trying to get some data to really nail that down. Some groups are locally hyper-diverse. We have a couple of species of clavulina around here. These nondescript-looking coral fungi that aren't as colorful as most of our ramarias, which are in a different genus. Never forget it. At our site in Guyana, we've got over 20 species of these coral fungi. And most of them, all of them except 2 out of those 20 are new to science. And we've described them all. We've really worked this group up, and this was a lot of work that Jesse was involved in. That's a typical clavulina morphology. That's one of our new species. Highly good edible mushroom. The natives eat it all the time. We eat it too. Branching coralloid pattern. Some of them are in clusters that don't branch much, they're monopodial. Then it starts getting weirder. Some species, like Craterelloides, are shaped like a funnel, like the genus Craterellus, which is in a completely different group, the Cantharellaceae. And then the weirdest ones we've found of all are these little resupinate cushion-like forms, Clavulina cerebriforma. It's just a little mound of tissue in clusters with basidia shooting off spores from the surface. So a high reduction clear down to an almost nondescript mound-like fruiting body, unknown anywhere else in the world in this genus. You get them under the microscope, all of these are good clavulinas. When you DNA sequence them, they all come out in the same genus for radical different very wacko morphologies. Some taxa that we've collected down there are previously described but appear to be regionally endemic. There's been some macro-mycological taxonomy work done, like in north-central Brazil by Rolf Singer, in similar ectotrophic systems south of Guyana. And when we find species that, by perusal of the literature, looking at herbarium specimens on loan, we realize we've got the same species in Guyana. Most of the time, it's something that Singer described from north central Brazil, like these. Cantharellus guyanensis, Ammonite campina, Zericonis amazonicus, Clavulina amazoniensis. All known from and described previously from collections in adjacent regions of Brazil or French Guiana, close to Guyana. When you get into the Cantharellaceae, though, of which Cantharellus guyanensis is, within that family, like compared to our Cantharellaceae around here, it's really weird. Because here we have one or maybe two species of the genus Craterellus, and they're nice funnels, highly consumable edible mushrooms. And most of our Cantharellaceae are good chanterelles, Cantharellus. Down in Guyana, it's flipped around. Everything's Craterellus, but there's only one Cantharellus species. And we've done taxonomy work on this. We've had to describe all of them except for Cantharellus guyanensis because it was previously described. And when we did phylogenetic analysis, all of our Guyana ones fell out in the Craterellus clade within the family. Only Cantharellus guyanensis falls out in Cantharellus. This mushroom is the most widely distributed species of ECM fungus known from South America so far, because I was able to turn up herbarium specimens from sites as distant as extreme eastern Brazil in the tropical region, and even in southeastern Brazil in the subtropical region down here. Material I collected in Guyana and French Guiana, and some Singer specimens from the central Amazon. We sequenced all of them. They're conspecific morphologically and at the DNA sequence level. So it's the only known example of fairly regionally widely distributed ECM fungus so far in South America. Some are radical new distribution records. This is Arthur Group. He recently finished grad student of mine, kicking back in the hammock. He must have finished all his work for the day, or he was slacking off again. I'm not sure when I took the picture. Sarcodon. This is a tooth fungus genus. We have several of them around here. They're prominent players in coniferous-dominated ECM systems in the rainy season, but essentially unknown except for a couple of records from the neotropics. We started finding sarcodons in Guyana and realized that other researchers that we knew had herbarium collections from other parts of the neotropics that they'd never worked up that ended up in this genus. So Arthur spent a lot of time describing new species in this genus from Guyana, Belize, Puerto Rico, and now Colombia from material we've got several others. A few are previously described taxa of broad distribution. These three different bolete species are morphologically dead ringers for species that have been described previously that have distributions that might span Southeast Asia, Central America, and Eastern North America, like Volatil sinatus. Austrobolitus rostrupii, a very distinctive looking bolete. That shaggy stipe, the mucilaginous pileus, is morphologically identical to the species that was described from Southeast Asia and Australasia. So we don't have comparative DNA sequence data to determine from a molecular standpoint if it really is the same species, but an absolutely thorough morphological examination, identical in every character, including the huge plethora of microscopic characters that you have to get when you work on these fungi. So we put the same species name on it until we know that. How diverse, though, when you go in and collect mushrooms in a forest, no matter how many times you go in there, how many years in a row, how many weeks in a row, every 2 days you go back in, collecting the same plots, keep logging off the species presence, you will never reach a point where you can go, I have found all of the macrofungal species that occur in this forest. This doesn't work that way. The longest plot-specific studies that have been done for the most continuous years are in temperate sites, up to 21 years of continual sampling in Switzerland is the longest example. They were still finding species in the plots on each successive year that they had never seen before in the entire sampling period. So you can't go in like you can pretty much with plants. You can sample and measure all the plants in a stand because their above ground portions are identifiable, stationary, and present all the but with fungal fruiting bodies, they come and go based on when the fruiting happens. So you have to go below ground and use molecular techniques to sample the roots with mycorrhizal fungi and see what you're getting from a DNA sequence identification standpoint in the root systems to get a better idea clearly of how totally diverse the system would be for that particular ecological group of fungi. So we've done some studies like that, looking at 3 different leguminous ECM plants that occur in the same forest over a localized area in Guyana. I'm going to gloss over some of these results so we can do the African stuff. And I'll just give it to you in a nutshell. You don't really have to look at the details of that bar graph. But if we look at the same exact plots, here's the bottom line, where we sampled the fruiting bodies continually for 8 years. We've continued to sample sporadically since then. We're up to 174 species known from fruiting bodies. When we sample below ground, you collect the mycorrhizae, hundreds or thousands of them, and DNA sequence each root tip individually. Very gargantuan effort to do it. You realize that approximately 50% of the fungi you're getting off the mycorrhiza themselves are species that you've never seen as fruiting bodies in the same exact plots of forest. We know that because we've got a DNA sequence library from all the fruiting bodies of those 174 species. We get the sequence, we can immediately check it against our in-house database and see if it's identical for that particular species level gene, so to speak. And if it's identical, that means we've recovered that species on a root tip. Well, when we do these kinds of studies, this one had 1,200 of those root tips sampled from 60 different trees among 3 species of host trees. Like 50% of them don't match anything in our local database that we know as fruiting bodies. That means there's at least 50% more species forming mycorrhizae in that stand that we've ever seen, with all this effort to go down there and sample every year for 8, 9, 10 years in a row. So if you figure in the unknowns from the roots, even in those design forests of the Upper Pataro, there's somewhere around 250 to 300 species of these symbiotic fungi in the stand, which is a figure that is very much comparable to diverse ECM fungal sites that have been more well studied than the temperate zone. So the idea that you can't have high diversity in this fungal group in the tropics does not hold when you look at the right systems. We also did this with one of the dipterocarps. I've mentioned this family before, and this is an interesting botanical aside because the Dipterocarpaceae, very dominant tree group family in the Southeast Asian tropics, Malaysian Peninsula, Malaysian Archipelago, Indochina. But they're only known from the New World tropics from 2 species that are known. And guess what? Both of them are in the Guyana shield. One of them is in Guyana, the other one's in eastern Colombia on the western side of the Guyana shield. So we've spent some time sampling this tree, which is Pachyrimea dipterocarpaceae, which is one of the 2 lone New World dipterocarps. It occurs in Guyana, and it occurs in a forest type that also has different species of assignments, so 2 actomycorrhizal trees together. But the dipterocarp distribution in the New World is very poorly known. There's either not very many of them, or we haven't found the rest of them yet, because Pachyrimea dipterocarpaceae is only known from eastern Venezuela with 2 different subspecies. One's in Guyana, one's in the adjacent part of Venezuela. And then Pseudomynotes tropimbosii was discovered only about 15 years ago in the western part of the Guyana shield in extreme southeastern Colombia. All the Dipterocarpaceae in the old world were ectomycorrhizal, and these, when you find them, both Pseudomynotes, the Colombian researchers determined that, we have the Pachyrimea, they're ectomycorrhizal in the New World too. But there aren't apparently very many of them. Are they a Gondwanan relictual remnant of the Dipterocarpaceae in the New World? Maybe. So these sites with the Dipterocarp, that looks, this is a 10-kilometer scale. Our main research area from all those other plots is in this part of the forest, and the dipterocarps are over there. That's maybe 30-some kilometers or so in a line. But there's a big mountain in the middle between our other research site, which is the closest place we can fly into. We can only get into the places that have airstrips close by. There aren't many of them in this interior Guyana. So we've had to trek up and over that mountain, which is that video that Jesse made is actually one of those cross-mountain treks to get over to the dipterocarp sites. And these are patches of non-forested savanna vegetation with fringing areas of forest that are rich in those Pachyrimaea trees. And there's Jesse holding a hanging on a mountain sign that we found in the bush. And I don't really have time to dig into the results, but basically, here it is in a nutshell. We found also plenty of fungal sporocarps under the dipterocarps, many of the same species that we found with Decymbi on the other side of the mountain, and some that so far seem to be unique to that dipterocarp system. When we sampled the roots, we pretty much found almost complete overlap in the fungi that are forming the mycorrhizae with species that we already knew, either as sequence root tip species or sporocarps from the Decymbi side. So we're starting to conclude that on a regional scale, even if you have phylogenetically different host trees, in this particular area, they're all kind of sharing the same common pool of mycorrhizal fungi regionally. That's kind of different from studies that have been done in other systems with eucalypts in Australia, some temperate systems with pinaceous and fugaceous hosts. You got an oak and a pine occurring in the same stand, and you adequately sample their root systems and identify the fungi forming mycorrhizae. There may be a very strong host-specific signal in the community and composition of ECM fungi on the roots of the oak versus the pine, even if they're growing in the same stand. Down in Guyana, it doesn't look like that. It looks like there are a few different lineages of ECM trees, but at least regionally, they're kind of all tapping into a common pool of ECM fungi because we don't see much host-specific variation between the fungi that we find on species A versus B on the host trees. So the African part comes in with a possible Gondwana connection. When the continents were all crammed together, the southern part of this big southern part of the continental mass was jammed together. This is roughly 230-some million years ago. You had a common landmass, really, of all the continents on the Earth, and a big chunk of the southern part of that landmass was basically called Gondwana. And from South American African standpoint, as the continents begin to separate, Antarctic and Australian stuff started going off down this way, South America and Africa began to separate. That's why to this day you can still put the two continents together like two pieces of a jigsaw puzzle. The Atlantic Ocean's been spreading and widening out, pushing those continents apart for about 100 million years. This is roughly the position of South America and Africa about 94 million years ago. They had a small, narrow strait between them. So it took a long time once those continents began to actually separate to get as far apart as they are today. So there was a long period of time where they were separated, but pretty close within the last 100 million years when they've gotten progressively further apart. Well, the Decymbi, forget about that phylogenetic tree, it's too complex to look at now. But Decymbi is this ECM alpinioid that's here now. And all of its closest relatives in the Fabaceae are in this part of Africa in today's world. The tropical parts, western central Africa, Congo Basin. So I've been hypothesizing for a long time that the Decymbi trees and their ectomycorrhizal symbionts might be a relic of a time when there was an ancestral lineage of this particular group of legumes that adopted this type of mycorrhizae that originated prior to the significant separation of Africa and South America by continental drift. So I've been wanting to go over there and start sampling the fungi in monodominant stands of some of these legumes in Africa so we can see if there's species level overlap morphologically, but especially get really robust molecular data sets so we can do phylogeographic analysis on the fungi to see if we can pick up signal that indicates that fungal taxa that are current now in today's world that exist on both sides of those continents that are in the same families, in genera for instance, if we can get evolutionary dates on those by phylogeographic analysis of their origin that predate the separation of the continents would allow me to say that it probably is a relictual situation in these systems between the two continents as we see it today. So I haven't gotten very far with that, but I did do one major expedition to the Congo Basin last year on National Geographic funding. So we're thinking Africa now. We basically, and that's hard to see, but there's a country map of Africa there. And the Congo Basin and the Guineo-Congolian rainforest that covers most of it is kind of like the African equivalent of the Amazon Basin. It's not as big, but there's a huge river, the Congo, big watershed, and most of the area drained by that watershed is lowland tropical rainforest, kind of analogous to the Amazon watershed in the South America. And the country of Cameroon is one of several countries that has part of its geographic region in the Congo Basin rainforest area. So we went to the Ja Biosphere Reserve, which is a fairly large protected area, 500,000 hectares in almost extreme southern Cameroon. And it's in the Guineo-Congolian rainforest realm. And in that forest, we already knew from prior studies, there were extensive monodominant forests of Gilbertiodendron weberi, which is a closely related seizout to Decembi in Guyana, phylogenetically, even though they're separated now by a huge ocean, etc. So we went down there, we got in, got a bunch of colleagues together from Cameroon, botanists and stuff, did an expedition down there, got into the Biosphere Reserve, and found beautiful stands of Gilbertiodendron monodominant forest, just a few miles into the park after we entered it. Extensive areas, big stands. Pods, the seedlings, very much exactly like Decembi. Not multiply reiterating stems of Gilbertiodendrons are single trunk and very large canopy trees. They're like 160 feet tall, which is tall by tropical standards. And huge dominance of the seedling and sapling pool, and lots of ECM fungi. So we got in there, set up a plot system. Oh, I threw a few botanical pictures that Todd Elliott took just so we could look at some plants real quickly. I didn't know any of the plants very well, certainly not at the species level, entering the African rainforest for the first time after being in the neotropics for so many years. But Todd, a student colleague of mine who's a professional photographer actually, Todd Elliott, took some nice pictures of some cultivated plants that the local indigenous people had in their little farms, like Theobroma, cacao. There's the cauliflorous fruits of the cacao plant developing on the stem in a small plantation. There's a very cute leguminous Caesalpinioid legume shrub with really beautiful flowers, Caesalpinia pulcherrima. This thing, though. Now, I'm fairly familiar with the Anacardiaceae. We've got some around here, like poison oak. Get down in the neotropics, there's a lot of anacard trees. Cultivated cashews in anacard, for instance, mangoes in anacard. There were these things, bush bonbons. We're walking around through Cameroon, guys, in the forest, and all of a sudden there's a small tree with these large, bright red, egg-shaped cauliflorous fruit clusters hanging off of it pretty much at face level. Easy to just reach out, and these are amazingly succulent fruits. They're super sweet, fleshy, juicy, and tart, all at the same time with a very thin, kind of fuzzy skin. They're easy to eat. And they're presented by the tree right at the exact level that if you were a terrestrial primate, like a chimpanzee or a gorilla, it'd be a piece of cake to walk up and rake these guys off the tree and gobble them down. And they're probably dispersed by terrestrial primates because there's chimps and forest or lowland gorillas in this system. But it was the most obvious presentation of a fruit for an animal to eat it I've ever seen in a plant. Because it's in forest understory. The top of the tree is like 50 feet up, but it's fruiting down here by the base with these really succulent bright red fruits. So we ate the crap out of these. That's Olivier Sinning, my botanist colleague from the National Herbarium who went in the field with us. These guys speak French, so they're, yeah, we call those bouche bonbon. Bouche bonbon. Yeah, and we saw, oh, those are pretty good. And then we started seeing them like every day out in the field on another tree. We were just gorging on these things. You couldn't eat enough of them. Your hands were all stained red and stuff, juice running down your mouth. And you could go, you know, it's probably about the same reaction that a chimp or a gorilla has when they find this. They go ecstatic and gobble them all down. It's an anacarp, we figured out. Just a few more plants. The Annonaceae is a big family in the tropics. We have some temperate pawpaws back east and stuff like that. But you get down neo and afrotropics, there's a lot of Annonacs, many of which have really good edible fruits. This is like a sugar apple type. This is a wild one, this Guiana confinus, that has these fruits that have separable compartments with a seed inside and very sweet flesh surrounding the seed. There's red stuff in there when you tear the fruit off. This Vitex, these things were for sale in villages and stuff. We never saw them growing wild, but they look like large Concord grapes. They're in the Bourbonacea, but they're woody, shrubby plants that bear a lot of these fruits that are cultivated or collected wild, that are just eaten out of hand, like dessert fruits, like grapes. We saw this thing, which is Fungi sanguinea. This is in a tropical group of root parasitic plants. Ballinopheraceae, I think they're restricted to the tropics, and they're not mycoheterotrophs like a lot of our Arcata chlorophyllus plants are around here. They actually tap into the woody root system of a tree below ground and suck nutrients directly out of the system by forming like a hostorium, like a mistletoe does on the above ground part of the tree. And then occasionally they'll throw up some kind of inflorescent structure above ground to do pollination and seed production and stuff.
I have a question.
Yeah.
Is there any relation to this in the Datura sanguinea?
No. What was the species that you mentioned? What sanguinea?
Datura.
No.
It's the only other sanguinea.
No, not related, because when you see a binomial name like this, the second part of the name, the species epithet we call it, there may be many species of organisms that have that same epithet. It's the combination of the genus name and the epithet together that gives that species its unique identity from a name standpoint.
I see.
So a lot of things are called sanguinea if they're red in color, because it means something along the lines of blood colored. So red things are often given a species epithet, something like sanguinea.
I got it.
So we set up hectare plots and camped out in that jungle in Cameroon for 2 months in the same spot in a bush camp, like you saw in the video. Had some Cameroon guys, some local pygmy guys, field assistants, and some Bantu villagers working in our team. Maylin Chin was there, she's in the crowd now with that iPad back there, and 2 other mycologists, Todd Elliott and a colleague of mine from Switzerland. And we set up hectare plots just like we did in Guyana, and we started sampling them for sporocarps of ECM fungi. I never did so much collecting and field describing in my life, and I've been doing this for years. Everybody was making field descriptions, but I had too many taxa for one person to work up each day. So just in a nutshell, in 3 hectares of Gilbertia dendron forest with 1 field season of sampling, that was 10 repeated rounds of systematic sampling in each plot stretched over a 7-week period. Came up with roughly 200 species of mushrooms in ectomycorrhizal lineages. Just that number alone is more than we've ever found in Guyana in 15 years of sampling in the Bayside system. And certain groups, much like Guyana, were dominant, like the Russulaceae, Russula lactarius. I never saw so many Russulas. You think you got a lot of them around here? Oh my God, like 52 different species of Russula in these pods. Morphologically different enough to say, well, they're probably different species. Just green ones, red ones, blue ones, everything. Lots of chanterelles. Believe it or not, somewhere around 20 different species of Cantharellus. 20 species of Cantharellus in these 3 plots and the immediately surrounding area. That's 20 different chanterelle species. So they were super diverse. Amanita was diverse, roughly 20 species. And boletes in different genera in general were very diverse, just from 1 season of sporocarp sampling. We collected a lot of mycorrhizal root tips and stuff, and we haven't even gotten to the point where we've done the DNA sequencing. So I'm trying to get multi-year funding to really expand our exploration of this system, so ultimately at some point we can get to the point where we can really make the definitive comparison of the fungal communities between the Guiana Shield and tropical Africa. In the meantime, we're definitely getting a lot of raw material for more taxonomic work. It's certain that a good deal of these African species are going to be new to science, and somebody's got to work them up. So there's, maybe I'll live to be 120, maybe I can get like 5 grad students at a time, each picking a group and describing 6 or 7 species for their masters, maybe. We'll see. But it's fun doing it because you just get blown away by the fungal diversity. These are like the 18 most common fungi sampled in our plots, pretty much representatives of Cantharellus, the Boletes, Inocetes. We've done, that being said, we've already cranked out some basic taxonomic work on certain fungi that we've got there. We've got a couple key collaborators that were keen to work on things like the Elaphomyces that we got. These little truffle guys. It's well known as Elaphomyces is in temperate regions and Australasia until we discovered 3 species in Guyana that were not known from the lowland neotropics. And believe it or not, to this date, there's not a single record for that genus in all of Africa, the entire continent. We found like 3 different species of Elaphomyces that were really common. We found repeated, made multiple collections in these plots in this area in Cameroon. We're working these puppies up right away because it's a new continental record for the genus. It's a major mycorrhizal group in a lot of other systems in the temperate zone, and they're poorly known in the tropics, if not completely unknown, as they are in the African tropics. And we got 3 nice distinct species that we're about 90% ready to submit the manuscript describing these. Interestingly, and I won't bore you with the details in the phylogenetic tree, but 2 of these species that have this grayish looking gleba, not this pink one, under the phylogenetic analysis, their closest immediate relative is one of the Elaphomyces species that we described from Guyana a few years ago. Across the genus, we have a lot of sequence data from temperate Elaphomyces and those that we have from the tropics. 2 of those Cameroonian ones within the genus come out as very strongly supported as most closely related to one of the species we described from Guyana, suggestive that they might have some evolutionary history. We found some new Inocetes in a different genus that Brandon Matheny's working up now, so we'll let that go. But just to finish off with this thing, I told you about Guyana gaster, this truffle armillaria from Guyana. Well, Rachel Koch is my colleague Kathy Ames, grad student at Purdue right now. She's been working on Guyana gaster stuff for a PhD down in Guyana. We found 2, maybe 3 different species of armillaria in Cameroon when we were there, and we collected them. Brought them back. I send the material to Rachel. She sequences the Cameroon armillarias and throws them into her big database of armillaria species and her Guyana gaster that she's working on from Guyana. And her current results is that the closest relatives of this truffle form of armillaria that we named Guyana gaster are the armillaria species in Africa, not the other armillarias that are in the New World tropics or the other armillarias that are in the temperate parts of the rest of the world, like North America or Australia. And this is a pretty solid phylogenetic analysis. So what it means, this group right here includes the African armillarias that we have sequence data from, including this one new species that we're describing from Cameroon, sequence data from Guyana gaster specimens from Guyana. They come out in a supported clade with very high statistical support as a distinct group relative to the rest of armillaria that occurs in various parts of the rest of the world. What this means is that however Guyana gaster got over to the New World and turned into a truffle somewhere along the line, its closest relatives alive in the world right now are its armillaria kindred spirits that are back in Africa, not the other armillaria good true armillarias that are in the neotropics or elsewhere in the Western Hemisphere. And what that also meant from a nomenclatural standpoint is that based on this analysis, we now have to erect a new genus for the African ones, because this type of result renders the genus Armillaria paraphyletic, we call it, because you got a distinct group of armillarioid mushrooms. They're guild, stipitate, regular mushrooms. They would key out to the genus Armillaria very easily morphologically. With sequence data, they are closely related to Guyana gaster, which we gave a different genus name based on its radical morphology, and no evidence at the time that it was nested inside of Armillaria, and should this be given the genus name Armillaria. But we can't keep calling the African Armillarias Armillaria now because of this result. So Rachel's proposing this new genus name for the African Armillarias as Paleo-Armillaria, which I really like the sound of that. It means ancient Armillaria. And that group is kind of basal to the rest of the genus when you look at the overall phylogeny. So these are the kind of things we work on, basically. But we wouldn't know any of this stuff if we didn't spend a lot of years going to really remote places and spending a lot of money on logistics and withstanding the vicissitudes of the environment, the rainy season. There's bugs everywhere. There's rivers flooding, washing out the camp. Can't get the specimens dry because it's too wet. But we do it every year over and over and over and over and over because we just keep discovering the most awesome stuff. The more we probe it and explore it, the more interesting it gets. And the more we start in our own little way to fill in that huge black box of what is unknown about the world's fungal diversity, especially in the tropics. So that was pretty long, but I warned you at the beginning. So I'll leave you with, this is one of Todd's pictures from Cameroon. We collect insect parasitic fungi all the time when we find them. We just keep stockpiling specimens. So we found a lot of these cordyceps fungi on insect hosts in Cameroon. This was a really spectacular specimen of a mature adult cicada that's dead as a doorknob, with the fruiting bodies of the cordyceps now coming out like two horn-like projections from the carcass of the host. Now, in this kind of situation, the soft tissues inside the cicada's body are completely gone. It's all fungal mycelium inside the exoskeleton, but the exterior portion is completely undamaged. And this looks like a frozen in time adult cicada, but it's been dead a long time, and all of its tissues have been converted to fungal biomass, and now the fruiting buds are coming out of this first. These do not attack people. So I hope they don't, because we collected a lot of them. I'm sure breathed a lot of the spores.
Well, thanks, Terry.
Yeah.
So, Terry, we'll linger for questions. How about we'll leave it at
That sounds
Okay,
fine.
cool. All right, thanks for coming.
Yeah. That is a native form of chewing tobacco, actually. But it's not just tobacco. The Patagonia Amerindians from that region they've been doing this since time immemorial. They grow a particular strain of tobacco and they harvest the leaves green. They swim out in a river when the water's low and collect an aquatic plant that grows directly on the rocks and get at it. They bring that aquatic plant in, let it dry out a bit, and then they burn it down to ashes in a pot for good luck. Strip the fleshy part off the nib, bring it back, mix it in